Hello everyone, and welcome to episode 15, Remastered. Today, I'm going to be continuing the series on genetics by exploring the structure and function of our DNA. Namely, I'm going to be exploring the vital processes of transcription and translation. In the previous couple episodes, I introduced the basics of genetics with Mendel's famous pea plant experiments, and then the process of meiosis. I feel like I've done a lot of talking about DNA without really ever going into detail on just what exactly DNA is, or how it's structured, or how it works. Well, all that is going to change today, as I dive into the immensely rich field of study that surrounds deoxyribonucleic acid. Today, you will learn about how DNA carries genes, and the biochemical mechanisms through which genes are expressed as proteins. If you listened to episode 4 on nucleic acids, you should be familiar with the basic structure of DNA, but for the sake of simplicity, I'm going to briefly recap some of the major information. Nucleic acids, like DNA, are biopolymers, composed of monomers called nucleotides. Each nucleotide is a ribose sugar attached to a phosphate group and a nitrogenous base. There's 4 different nitrogenous bases in DNA, which create 4 different nucleotides: adenine, guanine, cytosine, and thymine, also called A, G, C, and T. When in complementary strands, A and T can pair with 2 hydrogen bonds between them, and C and G can pair with 3 hydrogen bonds between them. The structure of DNA gives it directionality, possessing a hydroxyl end and a phosphate end, or a 3' and a 5' end, respectively. This is because the 3' end has a hydroxyl group on the 3' carbon, and the 5' end has a phosphate group on the 5' carbon. DNA is synthesized only by adding bases to the 3' end, but I'll be getting into DNA synthesis more in the next episode. In order for DNA to work, it must be expressed. So what does it mean for DNA to be expressed? As DNA has 4 different nucleotide bases, these are used as a code for specific amino acids, which form specific proteins. A sequence of DNA bases that codes for a protein is called a gene. It's very important to understand that not all genes are for protein coding. Many genes are regulatory, involved in coordinating and timing the expression of other genes, while others still seemingly do nothing, or they have a purpose that is not yet understood. As with DNA synthesis and repair, I'll be getting to the regulation of gene expression in a later episode. Today's episode is about the mechanism by which a gene is expressed. This is to say, the way that a protein is made from the nucleotide sequence in a gene. Now, this mechanism of gene expression has 3 parts, which are called, in sequential order, transcription, RNA processing, and translation. Chances are you've heard of transcription and translation. These are the processes through which a gene is read by a group of proteins to produce a complementary copy of mRNA, which is then read by another group of proteins to build new proteins. The rate of some of the steps in this process can be startlingly fast, like the transcription factor proteins that move along DNA at 50 base pairs a second to produce the intermediate mRNA molecule. So with this very quick summary, I think we can jump right into the meat and potatoes of the topic. I'll start with bacterial transcription, and then I'll discuss eukaryotic transcription. In bacteria, transcription begins when a suite of proteins, namely RNA polymerase, its subunit sigma, and some associated proteins called general transcription factors, when they all bind to the DNA and open up a sequence about 14 base pairs long. This small sequence of DNA contains a promoter region, called a promoter, which is a sequence that attracts the protein sigma and the RNA polymerase so as to initiate transcription. This is because, on its own, RNA polymerase is indiscriminate. It'll attach to any sequence of DNA and try to get started. But when RNA polymerase is in a complex with the protein sigma, the whole complex is much more selective in the DNA that it binds to. There's different kinds of sigma protein, and different kinds seek out different promoter sequences. Cells in different tissue types use different sigma proteins to access genes encrypted by different types of promoters. Once the RNA polymerase sigma complex binds to the exposed patch of DNA, it proceeds to continue to unzip the strands. On the DNA, the RNA polymerase moves in a 5' to 3' direction. That is, it moves towards the 3' end of the molecule and away from the 5' end. Sequences or base pairs are said to be downstream of a given point when they're closer to the 3' end. Those base pairs or sequences that are said to be upstream of a given point exist closer to the 5' end. So if we say that the first base pair in a bacterial gene is +1, then the promoter regions in the bacterial DNA, are somewhere upstream, around -10 and -35. This is to say that there's promoters around 10 and 35 base pairs upstream from the first base pair transcribed in a gene. In bacteria, these promoter regions are called the -10 and -35 boxes. The -10 box contains repeating thymine and adenine pairs, while the -35 box contains a TTGACA sequence, which is common in a wide range of organisms. Keep in mind two things. When they say -10 and -35, this is referring to a specific point upstream, but when they refer to the -10, -35 box, this is referring to a small cluster of sequences that exists in that general area. And then also keep in mind that the similar biochemical processes that are used to replicate and express DNA are as fundamental and as universal as you can get. All life shares this. This also touches on evolution, which I'll get into in more detail in the next series. Anyways, sigma is the first part of the enzyme complex to touch the DNA, binding between the -10 and -35 boxes. The complex opens the DNA so that both strands are accessible, and transcription begins at the +1 site. So the DNA is being funneled into this protein complex through the RNA polymerase molecule. And as the DNA is being shuffled through the inner corridors of these proteins, the DNA is being opened up. The strands are being split apart. One strand of this exposed DNA is called a template strand, because it's the strand that's being read by the RNA polymerase. This template strand is being used to create a complementary strand of mRNA, or messenger RNA. Now the other strand of DNA, the strand that's not being read by the RNA polymerase, this is called the non-template strand. It's also called the coding strand, because it contains the literal bases to code for the protein. The complementary mRNA molecule that's being constructed is identical to the coding DNA strand, because they're both complementary to the template strand. They're identical in the sense that they have the exact same nucleotides and the exact same sequences, with the exception of having the nucleotide uracil replace thymine. If I haven't said this before, there's actually 5 nucleotide bases. There's adenine, thymine, guanine, and cytosine, but there's also uracil. DNA only uses the first 4, but RNA only uses 3 of the 4 nucleotide bases that DNA uses. RNA uses the nucleotide uracil instead of thymine, and it pairs to adenine just like thymine does. So when everything is situated and transcription begins, the sigma protein will detach, and this will end the initiation stage. The second stage of bacterial transcription is called elongation. The emergent mRNA molecule grows by 50 nucleotide bases a second as the RNA polymerase reads through the template strand. New nucleotides are added to the 3' end of the mRNA molecule, causing its 5' end to protrude out from the RNA polymerase. As the DNA is being read, it's being moved through the RNA polymerase, guided by flaps and bumps and channels on the inside of the molecule. As the DNA helix enters the protein, a protrusion called a zipper will unzip the DNA, and both strands are guided through their respective corridors by a second protrusion called a rudder, and it's called a rudder because it steers them. And eventually, when the DNA leaves the protein, it gets re-zipped or resealed, and all of the nucleotide bases have their hydrogen bonds reformed. The DNA will leave the RNA polymerase in a helix, just as it entered. In another part of the RNA polymerase molecule, the growing mRNA strand is being guided through and out of the RNA polymerase. An intake cavity will suck up a stream of loose nucleotide bases that get incorporated into the mRNA strand. Suffice to say, the RNA polymerase is in full action during the elongation phase. DNA is moving through it at 50 bases a second, growing the mRNA at the same rate, and everything is going along grand. Until, of course, it all comes to a stop. The elongation stage ends, and the termination stage begins when the RNA polymerase reads a stop signal in the DNA. In bacteria, this stop signal in the DNA will produce a sequence in the mRNA that folds back on itself to create a hairpin loop. Just like when you take a string and you fold it back to run parallel with itself, you'll end up with a little semicircle loop at the end. In the bacterial mRNA, this hairpin loop basically interferes with the RNA polymerase and knocks it off the DNA. It's a mechanism that, in a manner of speaking, bumps it off the train tracks. Transcription in eukaryotes has many things in common with transcription in bacteria. After all, it's a very highly conserved system, genetically speaking, so it makes sense that even distantly related species would have many similarities. As there's many similarities and relatively few differences, instead of going over the whole process again, I'll just discuss the differences. First, eukaryotes have greater variety in their promoter sequences. This increased number of promoter sequences allows the cell to be far more selective when expressing genes. At -30, or about 30 base pairs upstream from the transcription start site, eukaryotes typically have a region called a TATA box, or a TATA box, which is literally a sequence of repeating adenine and thymine bases, or A's and T's. Second, eukaryotes use a series of proteins called basal transcription factors in place of the protein sigma. These basal transcription factors self-assemble into a protein complex at the promoter region, which helps to attract the RNA polymerase. This naturally introduces a third key difference. Eukaryotes don't use a single type of RNA polymerase. Instead, they use 3 different kinds. These are conveniently named RNA polymerase 1, 2, and 3, or poly 1, poly 2, and poly 3 for short. This gives eukaryotes another level of specificity when expressing genes. For example, RNA polymerase 2 exclusively reads protein-coding genes. The fourth major difference between eukaryotic and bacterial transcription has to do with the termination stage. In bacteria, a hairpin loop disrupts transcription. But in eukaryotes, the RNA polymerase reads a DNA sequence that's full of adenine, or A bases. This is called a poly-A signal, or a polyadenylation signal. Immediately after this signal is transcribed into the mRNA molecule, an enzyme comes in and snips the mRNA right before the poly-A signal transcription sequence. This will create a free-floating, newly made mRNA molecule. The RNA polymerase will then continue bumping along the poly-A signal until it reaches the end of the gene and falls off the DNA. Fifth, bacterial mRNA can be translated almost immediately after being transcribed. In eukaryotes, the RNA polymerase creates something called a primary transcript, or in the specific case of protein-coding genes, a pre-mRNA molecule. This primary transcript molecule has to go through a series of reactions before it becomes an active mRNA molecule that's able to be translated into a protein. This series of reactions must be performed because of the nature of eukaryotic DNA. In bacteria, the genes are continuous. An RNA polymerase can hop on a strand of DNA and print out a functional mRNA no problem. But that's not the case in eukaryotes. Eukaryote genes are split up by numerous little interruptions, sometimes hundreds, sometimes thousands. These interruptions are just sequences of normal nucleotide bases, but they don't code for anything useful or functional. When the DNA is transcribed, these interrupting sequences are also transcribed and incorporated into the primary transcript molecule. These interrupting sequences are called introns, as opposed to the sequences that actually code for functional stuff, which are called exons. So the eukaryote DNA has alternating intron and exon sequences, which are transcribed into the primary transcript molecule. To remove the introns and bind together the remaining exons, the cell performs something called RNA splicing. RNA splicing is very complicated, so I'll try to be as simple as I can. It's a process that requires two types of molecules. Some RNA molecules, called small nuclear RNA or snRNAs, and some proteins. Together, these molecules form what are called small nuclear ribonucleoproteins. The acronym for small nuclear ribonucleoproteins is snRNPs, which we can say phonetically as SNRPs. So RNA splicing requires a suite of SNRPs, and these target specific places around the exons and introns. Specifically, they bind to an adenine that's inside and near the edge of an intron, and they also bind to a 5' carbon that's between a guanine and a uracil at the boundary of the intron and exon. The SNRPs form a superstructure called the Spliceosome, which is typically a very large and very complex mass of interacting and interlocking proteins and RNA. The SNRP at the guanine-uracil interface and the SNRP at the adenine base are basically on opposite sides of the intron. When the SNRPs all associate together in the spliceosome, the intron gets pinched into a loop, with a little tail called the lariat, created by the excess RNA in the intron running past the adenine base. The SNRPs will cut the guanine and uracil bases, which breaks that GU interface. The SNRPs then drag the uracil on the cut end of the intron to the adenine base near the other end of the intron, and they bind them together. The intron has now been turned into a full loop, kind of like a lasso, with the circle forming between the uracil and the adenine, and the tail of the lasso being the little lariat. The guanine on the end of the first exon is brought over to the second exon still attached to the lariat tail. The lariat gets cut off, and the exons have their ends attached. The intron is then discarded with the spliceosome, and the new mRNA molecule is just a string of pure exons. I'll discuss splicing a bit more when talking about gene expression, where the cell splices in different exons to produce different mRNA from the same gene. As you might have figured, different mRNA codes for different proteins. And yes, this means that one gene has the potential to code for multiple different proteins. It's pretty exciting, but we'll get to it when we get to it. RNA created by RNA polymerase 1 and 3 would be ready for translation immediately after splicing is complete. RNA polymerase 2, however, creates RNA that has to be processed a little bit further. Protein-coding genes create primary transcription products that are spliced into molecules called pre-mRNA. This pre-mRNA is given an altered guanine molecule that it wears kind of like a hat. The guanine possesses 3 phosphate groups, and it has a core structure altered to that of 7-methylguanylate. This chemical hat is positioned on the 5' end of the pre-mRNA molecule, like a seal or a cap on the end, and so it's thus called the 5' cap. Furthermore, you have the 3' end of the pre-mRNA molecule that gets capped off with a tail of adenine bases that range anywhere from 100 to 250 bases long. This long string of adenines is called a poly-A tail, and the poly-A tail is not encoded into the DNA itself. The poly-A tail is added by enzymes after transcription. and both the cap and the tail are used to protect the mRNA from degradation, preserving their function and their accuracy. All of this, from the splicing to the 5' cap and the poly-A tail, are all a part of the intermediate step of RNA processing, which is the second overall step in the whole process. You often hear of transcription and translation without mention of the RNA processing in the middle, but this is likely because RNA processing only occurs in eukaryotes. It's relatively brief, and it takes place in the nucleus, in the shadow of transcription. So this takes us to the final step of the process, to translation, where the mRNA is read by ribosomes to synthesize the amino acid chains that will fold into functional proteins. Just like transcription, translation has 3 parts, with the same names: initiation, where the process begins; elongation, where the product molecule grows; and termination, where the process is halted or interrupted. The ribosome is another kind of ribonucleoprotein. Its 2 subunits are composed of both RNA and protein molecules. The subunits bind together, but each has a different role. The small subunit binds to the beginning of the mRNA molecule produced by RNA polymerase. The large subunit binds to the incoming tRNA and amino acids that will be used to make the protein. I'll discuss the tRNAs in a moment, but first let me clarify something about transcription and translation. In eukaryotes, transcription takes place in the nucleus. Once the mRNA is done being processed, it's shuttled out of the nucleus and pushed into the cytoplasm, where translation will occur. Bacteria don't have a nucleus, so their transcription and translation can happen in the same place, even concurrently with the same molecules. One can use an electron microscope to see the strands of bacterial DNA dotted with numerous RNA polymerases moving down its length. From these RNA polymerases, you can see the mRNA being printed out in long strands, which get longer as its RNA polymerase moves down the DNA. Numerous black clumps cling to this mRNA. These black clumps are the ribosomes, and they print the amino acid chains that fold into proteins. In bacteria, every step of this can be seen happening simultaneously. In eukaryotes, this process is discontinuous, as the mRNA must go through a brief period of travel to get out of the nucleus in between transcription and translation. Okay, so what about those tRNAs that I mentioned earlier? What are those things? Well, let me start by defining it. That'll help. So the T in tRNA stands for transfer. Transfer RNA is thus a type of RNA molecule that transfers stuff. In this case, the tRNA helps to transfer the amino acids floating around in solution into the growing polypeptide chain, or the growing protein. These tRNA molecules are relatively small. They're only 75 to 85 nucleotides long, with an L-shaped tertiary structure, and a secondary structure that's marked by beams of double-stranded RNA and loops of single-stranded RNA. It just so happens that the structure of the tRNAs gives them an amazingly crucial ability. A single-stranded loop in the tRNA molecule exposes 3 nucleotide bases called an anticodon, and a CCA sequence on the 3' end offers a site of binding for amino acids. So on one end, the tRNA can hold an amino acid molecule, and on the other end, the tRNA is presenting an anticodon in the form of 3 exposed nucleotide bases. To attach an amino acid to a tRNA, the cell uses an enzyme called amino-acyl tRNA synthetase. This enzyme catalyzes the phosphorylation event in which the amino acid is covalently bonded to the tRNA. Now there's 20 different amino acids, and each one has its own special amino-acyl tRNA synthetase molecule that catalyzes its binding to the tRNA. Sequential amino acids are bonded to the ribosome to produce a single polypeptide chain. So that's the amino acid, but what's the deal with the anticodon? You might be thinking, if these 3 nucleotides on the tRNA are called an anticodon, that must mean that there's a regular codon. This is a good line of thought, and the answer is yes. Yes, there is. A codon is a 3-base sequence on the mRNA. The anticodon is a complementary 3-base sequence on the tRNA. When the mRNA is being ran through a ribosome, the ribosome lines up the tRNA such that these complementary codon and anticodon sequences bind together. And remember that those tRNA molecules are associated with specific amino acids. In this way, the codons in the mRNA dictate the amino acid sequence in the polypeptide chain. But it's not quite so simple. Consider the numbers that are involved here. 20 amino acids, but 61 different codons? This implies that each amino acid can be associated with multiple codons. But how does this work? Each codon is composed of 3 nucleotide bases. The first 2 bases are very important, as these first 2 bases determine the identity of most of the amino acids. But the third base can afford a little variability. When positioned in the ribosome, the third base doesn't necessarily need to fit perfectly. The third base can be a little wobbly, so to speak, and still work fine. The third base doesn't really have to follow the strict AT-GC pairing. And in this way, 61 different codons are used to code for 20 amino acids, with each amino acid having, on average, about 3 different codons. I keep talking about how these molecules are positioned inside the ribosome, so maybe I should talk more about the structure and the function of the ribosome. Remember that the ribosome is composed of 2 subunits, the large subunit and the small subunit. The small subunit has a molecule of RNA, which binds complementary to the mRNA. Specifically, it binds to a start sequence near the 5' end of the mRNA. This start sequence is AUG, which stands for the adenine uracil guanine sequence that codes for the amino acid methionine. As a result of this, the vast majority of polypeptide chains begin with the methionine amino acid. The AUG sequence is typically 5 or 6 bases downstream from the ribosome binding site. So after the mRNA binds to the ribosomal RNA in the small subunit, the methionine carrying aminoacyl tRNA will then bind to the AUG codon on the mRNA. So we have an mRNA molecule that's being threaded over a small subunit of a ribosome, with a tRNA molecule bound to the AUG codon on the mRNA. If it helps you visualize it, think of it this way. Think of the small subunit of the ribosome kind of like a hot dog bun, and the mRNA is the hot dog that's running through it. A tRNA molecule coming and binding to the mRNA is kind of like an olive on a toothpick, and you poke the toothpick right into the hot dog. It's sticking up and out. Once this chemical structure has formed, then the large subunit of the ribosome comes and binds to the small subunit, completely enveloping the attached tRNA molecule. This will end the initiation stage and begin elongation. To continue the metaphor, this is kind of like if you're holding that hot dog with the toothpick sticking out, and you have a big hamburger bun come and be placed on top of it, and the hamburger bun will completely envelop that toothpick with the olive. And this is important because what happens inside of the large subunit, or inside of the hamburger bun in the metaphor, that's what binds these amino acids together and starts building this protein. I should mention that the ribosome has 5 major interior compartments. 2 of these interior compartments are like grooves or tunnels. One of these tunnel compartments is the space between the small and large subunits. The mRNA is run through this tunnel region as it's being read by the ribosome. The other tunnel region is at the top of the large subunit. Here, the amino acids are placed sequentially, bonded together into a polypeptide chain, and scooted out of the ribosome. Both of these tunnel regions run roughly parallel to one another. The other 3 compartments sit inside of the large subunit of the ribosome, and they connect the upper and lower tunnels together with vertical chambers that hold the incoming tRNA. These three vertical chambers are called the A site, the P site, and the E site. When the large subunit comes down and binds with the small subunit and completely envelops the tRNA and the mRNA, the tRNA that's poking up will fit right into the P site. It'll slide right into the middle, with its methionine amino acid positioned in the upper tunnel. During the actual process of translation, however, the tRNAs will enter the A site first. An incoming amino-acyl tRNA will enter the A site through diffusion, where its anticodon, if compatible, will bind with the associated codon on the mRNA. If it's compatible, the tRNA is held in the A site, and the amino acid that it's carrying is being held in close proximity to the methionine. At this point, the amino acids are bound together. This process causes the bond between the original tRNA in the P site and its methionine amino acid to break. So the methionine amino acid now shares a peptide bond with the amino acid of the tRNA in the A site, and it's not connected to its original tRNA. This second amino acid, however, keeps its bond to its tRNA molecule, for now. A group of proteins called elongation factors will then help the ribosome to move each tRNA molecule down one position. The P site tRNA goes to the E site, and the A site tRNA goes to the P site. So at this point, the E and the P sites are filled, the A site is empty, and we have a polypeptide chain that's just 2 monomers long. This process of sliding the tRNAs down the line of chambers is called translocation. These steps will repeat as the chain grows. A third amino-acyl tRNA will enter the empty A site. If it's complementary to the mRNA, its anticodon will bind with the next codon on the mRNA. Its amino acid will form a peptide bond with the proximal amino acid in the P site. This peptide bond formation will again break the bond between the amino acid and the tRNA that's sitting in the P site. Translocation will occur again. The original tRNA, the one that got pushed to the E site, is now ejected completely from the ribosome. The tRNA in the P site, freshly detached from its amino acid, is now shuffled into the E site. The newcomer, the tRNA in the A site who just added its amino acid to the polypeptide chain, is now shuffled to the P site. This process will continue, repeating again and again and again, adding a new amino acid each time. The growing polypeptide chain is directed out of the ribosome, where it begins to fold into its functional shape. Translation is terminated through the use of one of 3 different codons, called stop codons. These stop codons are UAA, UAG, and UGA. These are stop codons because there aren't any amino-acyl tRNAs that can bind with them, and when they show up in the mRNA, there isn't an associated amino acid that can continue the polypeptide chain, so the whole process stops. As a tRNA can't fill the A site if it can't bind to the codon, a protein called a release factor will step in to fill the gap. By filling the A site, the release factor is able to break the bond between the tRNA and the P site and its amino acid. This last action will free the polypeptide chain, and it'll slide out of the ribosome completely to form a fully fledged protein. After the mRNA has been read, the entire ribosomal complex dissolves. All of the subunits fall apart and disassociate and return to the cytosol. Now, the entire process is not quite finished yet. After translation, there's a few minor modifications that need to happen. These are called the post-translational modifications. Most generally, polypeptide chains need to be folded properly to ensure the synthesis of a functional protein. A misfolded protein doesn't work, and in the case of prions, they may actively cause damage. A group of proteins called molecular chaperones assist the polypeptide chain, making sure that everything is oriented and positioned properly so that the folding goes down without accident or error. This is a largely mechanical process that involves physically repositioning and moving the polypeptide chain. In episode 3, I talked about the primary structure of the protein, and how the primary structure determines the secondary structure, and how the secondary and primary structures determine the tertiary structure. Well, these molecular chaperones don't alter that in any major way, they just accelerate and supplement the natural folding to make sure that it happens quickly and accurately. These newly constructed proteins can also be given chemical tags, typically small sugar or lipid molecules. These tags serve as markers for other biomolecules in the cell, which tell them where this protein is supposed to go, or what it's supposed to do. Other tags are integral to the protein structure itself, and are necessary for its proper function. Another method of post-translational modification involves the addition or the subtraction of a phosphate group from the protein. Again, if you listen to episode 3 about proteins, you should have a general understanding of this point. Phosphate groups are very electronegative, carrying multiple negative charges on their oxygen atoms. This very strong polarity causes the phosphate group to react with the polar and charged side chains in the protein's amino acids. This interaction generally causes a significant conformational change in the protein. And as form follows function, the conformational change is necessary to activate or inactivate a protein. The regulation of proteins is often dependent on their coordinated phosphorylation, or dephosphorylation, which coordinates the total protein activity of the cell. Alright, well that's about it for transcription and translation. To finish the episode, let me summarize the whole process one more time. Transcription occurs when RNA polymerase works with other proteins, like the subunit sigma in bacteria and basal transcription factors in eukaryotes, to read the DNA and synthesize a strand of RNA. In bacteria, this mRNA can undergo immediate translation, but in eukaryotes, the RNA must be modified with a 5' cap and a poly-A tail before being moved out of the nucleus into the cytoplasm. Translation begins when the mRNA molecule binds with a small ribosomal subunit and an aminoacyl tRNA with a methionine amino acid. After these things associate, the large subunit will attach to the complex and begin processing the tRNA molecules. Incoming tRNA fit into the A site, where their amino acid gets bound to the other amino acids present in the ribosome. Then, all the present tRNAs are pushed down a space through translocation, and a new tRNA comes into the A site. Every tRNA molecule loses the bond to its amino acid at the P site, and they get discarded at the E site. This process is continually repeating, with each set of 3 bases on the mRNA coding for one amino acid to be added to the polypeptide chain. Eventually, all the amino acids are in place, and the polypeptide is finished. A protein called a release factor will fill the gap provided by a stop codon and terminates translation. A new protein has just been produced from a gene sequence. And with that, I think this episode is over. This was a very information-dense episode, so if you need to listen to it again, or listen to it while reading your textbook to soak in all the details, I think that's probably a good idea. And as always, thanks for listening.